Hello, everyone. Having finished the discussion of hemoglobin and the heme and the importance of iron in the heme for the binding of oxygen, which is essential for the functioning of cells, we turn now to iron itself, a very critical mineral, essential for the functioning of hemoglobin, as we said. So we're going to talk about iron metabolism, its deficiency, which causes iron deficiency anemia, and how we treat it. And finally, toxicity or overload of iron and what the consequences of that might be. So iron deficiency anemia is actually one of the most common anemias you'll encounter, the most common nutritional anemia worldwide. It is a common health problem and is particularly impactful on young children who are growing and who have need of iron, and on premenopausal women because, as you know, the menstrual cycle results in loss of blood, and there is a risk of developing iron deficiency from this blood loss. So they are particularly vulnerable. So if you look at the size of this problem or the burden of this problem, worldwide there are around 2 billion people with iron deficiency. Now, the fact that they have iron deficiency does not necessarily mean they have iron deficiency anemia. Only 500 million of them actually have anemia. So the fact is that without fortification with iron, food fortification of food with iron, the incidence of iron deficiency would be around 40% in preschool children, 30% in menstruating girls, and 38% in pregnant women. These are the three most vulnerable groups for development of iron deficiency and iron deficiency anemia. And the reason is that they are all in need of iron, particularly during these phases of growth, of loss of blood, and of course, pregnancy is a state of increased demand for all of these nutrients. So if somebody develops iron deficiency anemia and does not respond well to treatment with iron, or very importantly, I'm going to repeat this several times during this lecture, if iron deficiency occurs in men or in postmenopausal women, you have to definitely look for a pathology that is causing this iron deficiency. For example, one of the common causes of iron deficiency in men is colon cancer or GI problems that can cause loss of blood through the GI tract, or some worm infestations. So always look for for a pathology in those two groups. When you encounter it in the other three groups that we mentioned above, it is expected, and because there is increased demand for iron during this period of time. So iron is an essential metal, biometal. It has a number of chemical properties that allow it to react with proteins and with oxygen, as we mentioned before. And we said it is at the center of the heme ring and essential for oxygen binding. And we described how deficiency of heme, whether it's due to iron deficiency or other causes, will result in a microcytic hypochromic anemia because it is a deficiency in the synthesis of hemoglobin. And as we said, iron is also, and heme is also a component of other enzymes, not only of hemoglobin and myoglobin, but other enzymes. And therefore, iron deficiency not only has an effect on RBC function, but also on the function of other essential enzymes involved in respiration, in energy, DNA synthesis, and cell proliferation. So the symptoms of iron deficiency some may be ascribed to the anemia itself, and others may be ascribed to other problems resulting from this interference with the function of certain enzymes in the body. So in general, you can describe iron body stores in the following manner, and it differs between men and women. The most of the iron in the body is found in hemoglobin, as you can see, in myoglobin and enzymes, in transferrin, and transferrin is a special protein that binds iron in the plasma, and this is how iron is transferred from one site to another in the body. And then there is iron that is stored in the cells of the body in a form called ferritin. These all make up the body stores of iron. In males, it's around 4.5 milligrams per an 80 kilogram, 4.5 grams in an 80 kilogram individual. So iron is toxic as such. It can form free radicals. So there are special processes to handle it. It's absorption, its transport, and storage. And very importantly here, there is no excretory process for iron, in humans at least, and mammals. So it is not eliminated by the kidney, it's not eliminated by the GI tract in a specific manner. The way you lose iron is actually by losing cells that contain iron from the GI tract, from the genitourinary tract, from the skin epithelium, or by bleeding, because iron is mainly in the RBCs and the hemoglobin. So how do we regulate the levels of iron in the body? Normally you regulate something by controlling intake and controlling excretion or output. Here it is mainly through regulation of the absorption of iron from the GI tract. And of course there is therefore a very elaborate process of recycling of iron in the body. So there is a control of intake, control of absorption, and within the body it is preserved and recycled in various forms to allow the body to utilize iron as needed. If there's a deficiency of iron, then you would expect that the absorption of iron is going to be stimulated. If there's an excess of iron, you would expect that there will be an inhibition of the absorption. This is what we mean by regulation is at the level of absorption. And both the absorption and the regulation of the recycling in the body are under the influence of a hormone called hepcidin. This hepcidin is essential for maintaining total body stores of iron normal, so neither deficiency nor excess. So let us discuss the absorption of iron from the GI tract. Here on this slide you see the GI tract on this side. This is the lumen of the GI tract. This is the cell here, and this is the interstitial space where the blood vessels exist and will transport iron through the body. So what happens is that iron is delivered to the intestinal cells in two forms, either as a ferric form or as heme itself. In order to be absorbed, it can be absorbed as heme. When heme is absorbed, it is acted upon by heme oxygenase, as we discussed previously. Heme oxygenase releases iron from the heme, and the iron can follow one or two paths. It can either be stored in the form of ferritin here, or it can go to be absorbed, and we'll discuss this in a while. On the other hand, ferric form of iron cannot be absorbed as such. It has to be converted or reduced to the ferrous form, and that is done through an enzyme in the brush border of the intestinal cells called ferroreductase or ferrireductase. Once in the ferrous form, it can be transported by a special transporter called the divalent metal transporter into the cell, and therefore thereafter follows the same fate as the iron that was released from heme, which is either to be stored here or to be absorbed. Now, how is it transported outside the cell? There is a special protein called the ferroportin. Ferroportin transports the ferrous ion into the interstitial space, but ferroportin also is linked to a ferrooxidase, also called hephaestin. So these two are one protein almost, And in the process of transport, the iron is then converted to the ferric form. This is essential because for it to be transported in the blood, the iron has to be in the ferric form in order to bind to its transporter protein, which is transferrin. So now we go to the next slide, which shows the fate of iron beyond absorption. So on the left side here, this is the intestinal cells. This is what we described so far, and this is ferroportin here, leading to the formation of the green ferric. The ferric is then picked up by transferrin. It cannot be transported except bound to transferrin, special protein. Where does it take it? Well, it takes it to all the cells in the body. But of course, the target cells are the bone marrow, where the RBC production is taking place. These cells and other cells in the body have receptors for transferrin. So transferrin binds to the receptor. It's internalized with the iron into endosomes, which release then iron from transferrin, and the transferrin is then recycled back into the plasma, while iron is used for the formation of heme and hemoglobin. Other cells in the body can also take up transferrin, and for example, in the liver, transferrin is taken up and iron is released. Now, what happens to the iron? Again, it can either be used to form, to be incorporated into heme, that is important for other functions other than hemoglobin, other enzymes, or it will be stored in the cells in the form of ferritin. If it is to be recycled out of the hepatocyte, it goes through the same protein that exists on the intestinal basolateral side, which is ferroportin. This is the protein that transports it out of the cell back into the plasma and bound to transferrin. And the recycling of iron also involves, as we discussed in the lecture on hemoglobin, the fact that red blood cells, when aged, senescent, are taken up by the reticular endothelial system cells, macrophages in the spleen, that release hemoglobin, and then the iron is released. Again, the fate of iron is either storage, formation of enzymes of heme, or transport out of the spleen into the plasma to be delivered to the bone marrow. And this is a more detailed look at what happens in the cells that take up the transferrin receptor. Here, the transferrin molecule. This is the transferrin receptor. When bound, it gets internalized into the endosome. There are special enzymes here that release the iron. Iron in the ferrous form is used to make enzymes like cytochromes of the mitochondria, for example. Transferrin, ferritin as a storage form, or you can transport them back into the serum. So the transferrin that is free of iron is called apotransferrin. And these next slides simply repeat what I just explained. The ferroreductase is important for absorption of ferrous, or conversion of ferric to ferrous for absorption through the divalent metal transporter. HCP is a special protein. We didn't mention it, but it's depicted in the figure. That is the heme carrier protein that transports heme inside the cell, and the heme oxygenase breaks down heme to release iron. The export from the cells is ferroportin, which we mentioned earlier. We didn't mention mobile-ferrin, which is thought to be a protein that transports the ferrous from one side of the cell to the other. Hephaestin is the oxidase that converts ferrous to ferric, and apotransferrin binds the ferric to form transferrin, which transports it to the various cells in the body. The transferrin receptors exist and are essential for the uptake of the iron into the cells. The proton pumps in the endosomes release iron in the ferric form, and then this is reduced to ferrous by ferroreductase again within the cells of the body. And this is then transported from the endosome into the cytoplasm through the the divalent metal transporter again. And the transferrin receptor and the transferrin itself are recycled. So the iron, as we said, joins the body pool of iron, can affect heme synthesis and other enzymes, and the excess is bound as ferritin. This is the store of iron, which can provide iron for the functioning of the RBCs and other enzymes in times of iron deprivation. So even if you are not taking iron in through the food, you still have stores in the body that can sustain you for a while before you sustain or you develop iron deficiency symptoms of iron deficiency like anemia and other. The iron, as we said, ferritin is the form in which iron is stored. It is a protein that binds iron. Main sites are the liver, spleen, and the bone marrow. Hemosiderin is an aggregate of ferritin. When you have excess iron in the stores, you will see them as hemosiderin pigments in the bone marrow. They have a distinct color, a brownish color. Hepcidin, as we said, is the hormone that regulates this absorption and transport or recycling in the body. What it does basically is inhibit the function of ferroportin. And hepcidin can be up or down regulated. And as you would expect, when would you want to inhibit the function of ferroportin? When you have increased iron in the body or decreased iron in the body. Of course, when you have an excess of iron, because you don't want to keep getting iron into the system, otherwise you'll have an iron excess, an iron toxicity. So hepcidin is upregulated when you have excess iron and downregulated when you have iron deficiency. So the storage of iron, as we said, apoferritin is the form of ferritin which doesn't have iron bound to it. It binds iron to become ferritin. And as a rule, you should remember that if you want to assess the state of iron metabolism in the body and the iron stores, what you measure is ferritin levels in the plasma. Okay, ferritin is in the stores, in the tissues, but it is in equilibrium with ferritin in the plasma. So if you measure ferritin in the plasma, you can get an idea about the total body stores of iron. And that is what always must be done in order to ascertain that there is iron deficiency and that the anemia that you see is due to iron deficiency. So as you can see, ferritin contains 37% iron. That is 4,000 atoms per molecule of ferritin. Hemoglobin, remember, has 4 atoms of iron per molecule. So see the difference. So hemoglobin is a good transporter, but not a good storage site. Ferritin is a good storage site for hemoglobin, for iron. Transferrin, on the other hand, only 2 atoms of iron per molecule. And as we said, hemosiderin is this hemoglobin-derived pigment that consists of aggregates of ferritin, along with lipids and other membrane constituents. So, how do we regulate ferritin, transferrin, and iron in general? So when you have low iron in the body, low iron deficiency, there are certain proteins called iron regulatory proteins that can bind to elements or sites on the messenger RNA that regulate iron expression or expression of these proteins. So for ferritin, they bind at the 5' end and will block the translation. Whereas for transferrin, they bind at the 3' end and stabilize the messenger RNA, which means that maintain production of transferrin receptor. So when you have iron deficiency, what do you expect to see? A decrease in ferritin. because obviously you don't need ferritin because you don't have enough iron in the body to store. But you need excess transferrin receptor so that you can utilize this iron. Whatever iron is available, you can take it up into the bone marrow and use it to make RBCs, and take it up by the cells and use it to make whatever is essential. On contrast, when you have high iron levels, there is the opposite happens. So there's no binding to the iron response elements or regulatory proteins, and less formation of transferrin. That's why, and the reason is you want to limit the uptake of iron. But you need more ferritin to store whatever iron is in the body, with the excess iron that is in the body. And this is the slide depicting this. So in the absence of iron and in the presence of iron, the absence of iron, the iron regulatory protein binds to the iron response elements and blocks the transcription of ferritin, whereas when you have excess iron, you have the opposite. I'll just read this and and it will explain it. And as we mentioned before, the main regulator of iron levels in the body and in the circulation is ferroportin. Ferroportin, the protein that transports iron from the cell into the interstitial space and into the blood, which exists in all the cells which utilize or store iron. So what regulates ferroportin? We said it is hepcidin, the hormone that's produced by the liver. And basically hepcidin binds to ferroportin, phosphorylates it, or results in phosphorylation, internalization, and degradation. So basically hepcidin production decreases in iron deficiency because you don't want anything to inhibit ferroportin absorbing sodium from the intestinal tract and making it available to the bone marrow by releasing it from the internal stores of the cells. So in iron deficiency, hepcidin production is inhibited, whereas hepcidin production is increased in situations where you have excess iron in the body. You want to limit the availability of iron. You don't want to absorb iron anymore. Remember, there's no way of getting rid of iron from the body except by sloughing of these cells from the GI and the GU and the skin. So here you want to decrease the absorption of iron. It's also upregulated during inflammatory processes. So as a result, let's move on, and I'll be asking you a question very soon. So enterocytes, here are the enterocytes which are releasing or absorbing iron into the circulation. These are the cells in the spleen or wherever that also release iron from hemoglobin and can store it and then release it when it is needed into the system. And hepcidin, which is produced by the liver, can block both the absorption and the internal release of iron into the circulation. You get anemia that resembles iron deficiency anemia, but in fact, it may not be iron deficiency. So this is what we've been talking about. You're shutting down here and here and here. Ferroportin is being inhibited. And so these bone marrow cells are not able to get the iron delivered to them. And so when do you get this chronic impulse to shut down ferroportin? When you have a chronic stimulus for hepcidin release. And one of the things we talked about is chronic inflammation. And I think you have been exposed to the fact that chronic inflammatory conditions are associated with an anemia. And the reason for the anemia is, to a large extent, this mechanism: the release of hepcidin, decreased ferroportin, decreased transport of iron, and iron is not made available to the bone marrow in enough quantities and may result in anemia. Now, of course, this mechanism is also a way of getting rid of iron from the body, and that is by iron remaining in the intestinal cells as ferritin. And this gets lost from the body when the cells slough because they cannot be absorbed. They remain there, and eventually these cells are lost. Disruption of the regulation of hepcidin, as we said, results in a deficient response to Fe loading, right? So if you have iron overload, you expect that hepcidin release will increase. But there are situations when there is disruption of this release, of this regulation, and there is not this response does not happen. And in this case, what do you expect will happen? Obviously, you cannot control the input of iron anymore. You cannot reduce it, and you will be overloaded with iron. And this is a condition that is known as hemochromatosis, can lead to hemochromatosis, where iron is excessively deposited in the tissues of the body, and this can lead to organ damage and disruption in organ function. On the other hand, hepcidin release is decreased by obviously iron deficiency or hypoxia, which is interpreted as iron deficiency, or when you have simply you're losing blood or removing blood from the body. This decreases hepcidin release, and as a result, you increase iron absorption and the function of ferroportin. Okay, so how much iron is needed by a normal human being, man or woman, or at various ages? Well, there are two main determinants. First is the need for growth, and the other is the replacement of whatever physiological losses are taking place. For men, the needs are approximately 1 milligram per day, whereas menstruating women, because they're losing blood, require a little more, 1.54 milligram per day, whereas pregnant women require much more because the fetus needs also iron, so it's 5 milligrams per day. So what are considerations when you think about whether we are meeting our daily requirements? Obviously, the kind of food that you're eating, its content of iron, and its availability or bioavailability. Where, for example, heme iron is only 6% of dietary iron, but it is 30% of absorbed iron, because heme iron is more bioavailable than inorganic iron. So a vegetarian diet is associated with poor absorption because you don't have heme in vegetables. And the diet, actually, the vegetarian diet may contain some inhibitors of iron absorption. If you add meat, of course, you're going to get better absorption. And it has been noticed that vitamin C also enhances the absorption of iron, elemental iron. And this slide shows you in a table what are the factors that favor absorption and factors that reduce absorption of iron. We discussed some of them, like the heme iron, the ferrous form, presence of acids like vitamin C, and so on, decreased hepcidin levels. Meat obviously is a better source than vegetables for iron. And iron absorption usually, under normal circumstances, around 5 to 10% of the total iron presented. And these are some of the sources for vegans. So we said iron is present in animal products, meat, and so on. But if you're not a meat eater, then you have to resort to these kinds of foods: chickpeas, hummus, lentils, etc. Spinach, which I'm sure you're familiar with Popeye, the sailor man who once he eats spinach becomes very strong. The idea is that because it has iron in it. Well, the fact is that the iron in spinach is not the best source of iron for the body, because there are other products in the spinach which can interfere with the iron. absorption, and it's not in the form that is best absorbed, as you know, which is heme. So men, on average, as we said, they take in 15 milligrams per day, and the absorption, as we said, 5 to 10%, usually 6%, gives them 1 milligram per day. That will be enough for a normal functioning man with no pathology. Women, because they have a higher absorption rate due to the fact that they have slightly lower hematocrit, will take in 11 milligrams per day to give them 1.5 milligram per day, their daily need. This is menstruating women. Obviously, iron deficiency is going to lead, as we discussed earlier, to a more efficient absorption, going up from 10% to up to 20% for heme iron. And this graph shows you the relationship between the iron stores in the body and the absorption of efficiency of absorption of non-heme iron. So when you have high stores, 1000 milligrams, the absorption is around 6% or 4%. And as you deplete iron and decrease the iron stores, the absorption of iron is increased, the efficiency of absorption is increased. And this is an important slide because it shows you the people who are in a precarious situation, who are at risk of developing iron deficiency, because it relates the iron requirement to the available iron in a typical diet or a typical intake by any of these. So, for example, infants' requirements around 67 microgram per kilo. If they are eating normally and in a regular diet, they can take up to 33 to 66 micrograms per kilo. So they are a little on the negative side. There's not much safety here. Similarly, for adult females, where the available iron conceivably can be less than the requirement, and definitely for pregnancy, and that's why pregnant women have to have supplements of iron during pregnancy.